particular picture. So as we are looking at membrane potential, this is going to be an incredibly important thing to make sure that you understand. The word potential, if you think about potential, that means that somebody or something has the ability to do something. So membrane potential, meaning that somebody or something has the ability to do something. What we're looking at here, specifically for the neuron, is that we're looking at the ability for a neuron to take an action. So that is our membrane potential. We'll be talking about how the neuron's membrane is going to affect its ability to act. So whenever we look at the resting membrane potential, sometimes abbreviated RMP, you definitely want to remember that the resting membrane potential means that that neuron is in a restful state and it's actually just sitting there. It's not actually doing anything at that particular point in time, but it has the perfect ability to. So if you think about like a kid who's in high school and maybe they have great potential, but if they're just sitting there and they're not utilizing their potential, then they're not going to be able to reach their full value, right? So whenever we're looking at the neuron, this is it in its state of rest. Now, if there is some sort of stimulation, whether it's external or internal, that stimulation can then cause that neuron to move out of a state of rest and to start making some changes within its membrane. So if we have a graded potential, your keywords here are going to be temporary and localized. So make sure you circle temporary and localized, because this is going to be the important thing to remember about a graded potential. It's not long lasting and it doesn't stretch the entire length of the neuron. It's just going to stay in one particular spot and come and go pretty quickly. The action potential, the big focus of today's lecture, is the actual electrical impulse. Now, this is going to first start with that graded potential, but what we're looking at is we're looking at a signal that is going to be sent along the entire axon. So this is going to go propagate, mean to move, along the surface of the axon all the way to the synapse. And so we'll learn today where the synapse is and what's going to be happening at the synapse. So if you need your definition, this is the definition for action potential, okay? The electrical sequence of events that allows a neuron to communicate with other cells. It can communicate with other neurons, it can communicate with glands, it can communicate with muscles. This is the neuron's way of communication, is through these action potentials. So last time we drew a picture and it basically looked like this. So you can see here, this is the cell body or the soma. These are the dendrites. Remember, for the dendrites, we underlined the R because the dendrites receive information. And then the axon is that long nerve fiber that's going to send the information away from the soma and towards the synapse. So we underlined the A in axon because the information is going to travel away from the soma and only in one direction. Very important to remember that it only goes in just that one direction. So for today, new stuff. Whenever we are looking at cell membrane potential for a neuron, we are going to say that the neuron is polarized. Okay, very important word, polarized. That means that the inside of the cell is going to be differently charged from the outside of the cell. So take out your picture that you were drawing, and what I want you to do is I want you to put little negative signs inside of your cell, okay, and then positive signs on the outside of the cell. So we have two different charges, which is going to make this a polarized membrane. Now, the inside being more negative, the other thing I want you to do is I want you to write down a little mnemonic on the inside of the cell, PIN. Okay, PIN, pin. And what this means is you have proteins, That's one of your P's. You have phosphate, that's another P. And you have potassium. These are all inside of the cell, the 3 P's, proteins, phosphate, potassium. I is for internal, these are inside the cell. And N is for the charge inside the cell is negative. So 3 P's internally make this a negative charge. Okay? Polarization is going to be very important to allow us to have those action potentials and allow the nerve to actually, or the neuron, excuse me, to communicate with other cells and tissues in the body. So that's why it's very important you remember the inside is negative and then the outside is going to be positive. So what I'm going to do here is I'm just going to make sure that we're all on the same page. So I'm going to draw a little neuron, kind of like I did the last time. Let's not use that one. Let's use this one. Okay. So what was this cell body called? What's an AKA for the cell body, you guys? Huh? Soma. So Soma is the AKA for cell body. So the whole thing that we draw is going to be called the neuron. What is this long thing right here called? Axon. Axon, perfect. So what are these things called? Dendrites, very good. So our dendrites, what do they do? Receive, perfect. And so the information is going to be received at the dendrites, taken through the soma, and then sent along to the axon. Anybody remember what these extensions at the end of the axon are called? Telodendria. Yeah, I hear someone trying to say that crazy word, huh? Telodendria. Okay, and then what's at the end of the telodendria? Synaptic terminals or axon terminals. Very good. Okay, what do we have inside of the synaptic terminals? The neurotransmitters. Perfect. Somebody has been studying. Excellent. Okay, so what I'm going to do is go ahead and add some text to this. What I said to put inside was PIN. Remember, that is representing proteins, phosphate, and potassium are internal, and the charge inside of the cell is a negative. So let me put some negative signs. Okay, outside of the cell, we have a positive charge. And what you're going to find outside of the cell that's really helping to make this positive charge is going to be sodium. Okay, so let's put sodium outside of the cell so that we know that that is going to be the primary ion out here. Potassium was our primary ion inside. I'm going to go ahead and put, remember the signal for potassium is K. Even though it starts with a P, remember it's K. And potassium is positively charged. So don't let that throw you. Potassium is positively charged, but the inside of the cell is negative because of those proteins and those phosphates. So the summation of all of the charges internally makes it a negative charge. Okay? So whenever we are looking at our neuron, the other thing that I had you guys draw was the axon hillock and the initial segment of the axon. So we'll come to that here in just a minute and its importance in these action potentials. Let's see if I can. Oh, now it's going to be on top of it. Oh, that's interesting. I don't want to like that. Okay, so as we are looking at the sodium and the potassium ions, it's important that you circle this word, move, because these things are going to move across the cell membrane according to diffusion rules. So diffusion means that something is going to move from an area of higher concentration to an area of lower concentration, okay? If, for instance, there's like 100 people inside of this room and there's nobody out in that hallway, it would be natural for some of us to want to get up and actually go into the hallway where there's more space to spread out. So that's what ions do as well. They want to move from an area where there's a high concentration to an area where there's a low concentration, so they can kind of spread out. Now, the resting cell membrane is more permeable, circle that, more permeable to potassium than it is to sodium. And what that means is it's more likely for potassium to actually leave the cell and go out into the extracellular fluid than it is for the sodium to come from the outside and work its way in. Because of this, what that means is we've got to have something to counterbalance this. And so what I want you to draw on your picture is a potassium and sodium pump. So maybe just put like a little circle on the cell body and just label it the sodium-potassium pump. The sodium-potassium pump is going to be very important because this is going to help move sodium and potassium so that we can maintain the normal resting membrane potential. Okay, so you want to put on your picture the pump, draw a little arrow that shows that it's going to transport 3 sodium out and 2 potassium in. So arrow out on the sodium, 3 of those come out, and it'll draw 2 potassium in. The reason why this is important is because remember we have to have the polarization. So inside of the cell, this resting potential has to stay at negative 70 millivolts. If we were to measure it, it must be negative 70. So that's what you see down here. This is a very important number to remember. Negative 70 millivolts, that's our resting membrane potential. And because that potassium has an affinity to leak out of the cell, we need the sodium-potassium pump to help maintain that negative 70 millivolt charge in the cell, in the neuron, because if it's not, then it's going to cause problems with action potentials. Okay, so whenever we're looking at that cell, there are these things that are called channels. These ion channels are very important because they will regulate the passage of specific ions across that membrane. Make sure that you see here, some are always open. That potassium that will naturally come out of the cell will go through an open leak channel. So you may see it referenced as passive or you may see it called leak. Either way, it's the same thing. So this leak channel, it stays open, which is why potassium can actually leave the cell. Other channels are going to be carefully controlled, or what we would refer to as gated. So it will have this gate-like mechanism, and it will only open under certain circumstances. And that's basically what we're going to be talking about today, is what are those circumstances that will cause the channels on the neuron to open so that we can have this movement of sodium and potassium that will allow an action potential so the neuron can communicate appropriately. So on your picture, up in the top corner, you need to write channels, okay? Because that's one of the big, huge parts of understanding the neuron is you have to understand the channels. There are multiple channels, so make sure that you write underneath where you wrote channels. You have a passive channel. This is the leak channel. This is the one that's always open. And you can see here that it's located on the entire neuron. So we have it on the dendrites, we have it on the soma, we even have it on the axon. So these passive channels are everywhere. The chemically gated channels, also known as ligand channels, are going to be on the dendrites and the cell body. The dendrites and the cell body, you can see that these are not along the axon. The voltage gated channels are at the Axon Hillock as well as along an entire unmyelinated axon. If the axon has myelin, it will be at the nodes of rhombi instead of along the entire axon. So this is where the axon hillock becomes very important, because the axon hillock is going to be a space where you have those voltage gated channels and those chemically gated channels kind of all in the same area that are going to determine whether or not we're going to get an action potential. So what you see here, function, voltage gated channels, action potential. These other channels are not going to give you the action potential. So we have to have the voltage gated channels opening up in order for there to be an action potential. Okay. There's also mechanically gated channels. These are going to respond to any distortion of the membrane. So they're found in sensory receptors. So for instance, if somebody were to pinch you on the skin, that distortion of your skin would open up those mechanically gated channels. That's why they're found in sensory receptors, because it's all about physical change. Whenever we're looking at this picture up here, this is just a recap, just to show you positive on the outside in the extracellular fluid, negative on the inside. What was your little acronym that you wanted to write on the inside to help you remember what's in here? PIN, exactly. So PIN is your proteins, it's your, what else? Phosphate and your potassium, perfect. So internally, that makes for a negative environment inside of the cell. Remember, this is called polarized because we have the opposite charges internally versus externally. Resting potential, what is our measurement for our resting potential? That number you don't want to forget. -70, perfect, -70 millivolts. If we have some sort of stimulus, let's say if we walk outside and there's a little gentle breeze, that little gentle breeze, the neuron is going to be aware that something has changed in your external environment. So what it's going to do is it's going to create this graded potential. Okay, it's aware that there's something different, there's something going on. The graded potential, there was 2 words that I told you to remember. What do you want to remember about graded potential? It is temporary and it is localized. Okay, temporary and localized. The graded potential, if there is enough stimulus, can potentially produce an action potential. However, what you see down here, this little note, the stimulus must be powerful enough to open up the voltage-gated sodium channels. And we'll talk about that stimulus getting to a certain level to allow those channels to open and give us an action potential here in a few slides. So if we have an action potential, this will then trigger some synaptic activity. And the synaptic activity means that the neurotransmitters from that neuron can then be passed on to the next neuron or the next cell in line. And we'll also walk through that so that you can see how that happens as well. Okay, so here's a good chart that will help you differentiate the graded potential versus the action potential. We're not going to spend a whole lot of time in graded potential since the big gist of this all is going to be about action potentials. Okay, so that's our big thing here, is that we want to be able to see how is the neuron going to actually communicate. So here, this little note up in here in the red, you want to make sure that you put a little star next to that, because the axon hillock, if you did not draw it on your neuron already, you want it to be on your neuron because this is what we call the trigger zone. So the trigger zone is the area where we will have those chemically regulated gates and those voltage regulated gates kind of meet up so that we can get that action potential going. You can see here that, as we said earlier, the cell body and the dendrites is where we would have ligand gated channels. along the axon, and in the axon hillock is where we're going to have those voltage gated channels specifically for our sodium and our potassium, since those are the key players helping us to make this action potential. But now you need to add also to your picture, down at the end, in the axon terminal, a voltage gated calcium channel. So you've got multiple channels on your picture. You've got lead channels, you've got ligand channels, you've got voltage gated channels, and now you've got these calcium channels that are also voltage gated.
The voltage gated is the neurotransmitters?
The voltage gated channel is the one that will only open under a certain stimulus. The one with the neurotransmitters is going to be specifically the calcium channel. Okay? Okay, so whenever we say that neurons are able to respond to any changes either in our environment or within our body, we say they are excitable. Okay, we have the ability to excite them based upon stimulation from various things, either externally or internally. Whenever they get excited, they're going to change their resting potential. What was the resting potential? Negative what?
70.
Negative 70, very good. If they change their resting potential, what's going to happen is we get voltage gated sodium channels that are going to cause an influx of sodium into the cell. That stimulation can open up those voltage gated sodium channels and allow sodium to come from the outside to the inside. If that happens, if sodium, which is positively charged, comes from the outside and goes to the inside, what's going to happen to the charge on the inside of the cell? Is it going to get more negative or less negative? Less negative, okay? Or more positive. So whenever we bring in a lot of positive ions into that negative environment, then we're going to change that negativity into a more positive state. When we do that, what we're saying is this is depolarization, a very important word, okay? Depolarization means that the inside of our cell is now becoming more positive. It's no longer at negative 70. If we are able to get to the point that we get to negative 55 millivolts, we call this the threshold. So circle that word, threshold, because that negative 55 means we are now going to get an action potential. It's not going to happen, the action potential won't happen if we're negative 63. It's not going to happen if we're negative 57. It has to be negative 55 in order for us to have an action potential. Anything less than that is only going to give us the graded potential. So if we have a strong enough stimulus, then we'll have enough sodium ions that will come in through their voltage gated channels, raise it to negative 55, and now we're off to the races. Now we can do the big stuff. So whenever we're looking at this picture here, what I want you to notice is you can see these are your chemically gated channels that we talked about. Remember, the chemically gated channels are on the dendrites and they're on the cell body. And what's happening is you have this neuron. Let's pretend that there is an entire drawing over here of this neuron. All you're actually seeing in this picture is the axon terminal, okay? So you have axon terminal from these different neurons. These little things that you see here are the neurotransmitters that are being emitted from this axon terminal, and they're going to be received onto the dendrites of this neuron right here. When those neurotransmitters are released, it causes the sodium gates to open and sodium can rush inside and make this a more positive charge. However, what is this word, or not word, what is this number? Negative 62. What did we need in order to get an action potential? Negative 55, exactly. So this does not give us an action potential. There's not enough of a stimulus to cause enough sodium to come into the cell. So this gives us not an action potential, but a what? Starts with a G.
Graded.
Graded, perfect. So this is our graded potential, okay? Now, if we look at this picture, you can see here that we do have enough sodium entering in to these chemically gated channels that it actually causes these voltage gated channels to open up here in the trigger zone. Remember, the Axon Hillock is the trigger zone. And so now what is this number? -55. That is our threshold. So that's what it's showing you on this graph over here. This dotted line means that the stimulus was strong enough to get the neuron to threshold. And what happens after that is very, very fast. So we have an immediate upswing on this graph in a positive direction. So when we move in the positive direction, that means we're moving away from polarization. So right on this graph, this side right here, you want to write depolarization. Okay, that depolarization is movement from -70 up to +30, +40, depending upon which textbook you read. Okay, so what goes up must come back down. So what you see immediately after depolarization, after we tap out at that +30, +40, what you're going to get is repolarization. So over here on this side of your graph, on the downward trajectory, you want to write repolarization. We're going back to being polarized. So repolarization is our return to the resting membrane potential. They all start with RE. Repolarization, RE. Return, RE. Resting, RE. So re. Not like Rihanna, but you know, if that helps you remember, then whatever it takes. So this is your repolarization. Now, what was our resting membrane potential measurement? -70. Right, so now what you're seeing here is that this is actually dipping a little bit below -70. So right here, what you want to write in on your graph, the fact that this came below -70, hyper, H-Y-P-E-R, hyperpolarization. So we actually went too far. Okay, we went too far. And I'll explain in a minute why that happens and how we get to hyperpolarization. But as you can see, ultimately, it's going to come back up. It takes some time, but eventually it's going to return back to that -70 of the resting membrane potential because of those potassium leak channels and because of that sodium potassium pump. It will help with reestablishing that resting membrane potential. So again, an action potential is the electrical sequence that allows the neuron to talk. We have to have the action potential if our neurons are going to communicate. There's no reason to have neurons that cannot communicate. That would not make for homeostasis. So we want to make sure that all of these things work exactly as they're supposed to, and that the parameters are set very, very tightly for that neuron to be able to have an action potential. That's why it's important we remember -70 is our resting. We have to get to -55 in order to reach the threshold. And then we're going to go up in a process of depolarization to a +30 or +40, come back down to repolarize. But if we go a little bit too far, that's our hyperpolarization, and ultimately we end up back to our resting membrane potential so the neuron can do it all over again. Okay, so it's the changes in the RMP, the changes with the sodium and the ion, or the sodium and the potassium ion movements that are going to allow all of this to occur. so that that neuron can communicate. So look at this picture right here. What I want you to see is this is all sodium on the outside. This is all potassium on the inside, okay? When those voltage-gated sodium channels open, sodium comes rushing into the cell, causing it to be less negative or more positive. Whenever we look at the graph, what you're seeing here in this area of depolarization is sodium channels that are open, thereby allowing the sodium to come into the cell, while potassium channels are closed. So very important, put a little star next to that you remember during depolarization time, the sodium gates have to be open, potassium closed. Now, in order for us to go back to repolarization, what has to happen is those sodium gates have to close. Those sodium gates will close off and not allow any more sodium to come into the cell. And then once those sodium gates close off, potassium gates will open up and it allows the potassium to leave the cell. So we're making all of those positive charges leave the cell, which is then going to make the inside go back to negative. Okay, so that's why you have this over here during repolarization. Sodium channels are closed, potassium channels are open. Now, I told you I was going to explain the hyperpolarization. So this is what you have to write down on your picture down here for the hyperpolarization. The potassium channels close very slow. So because the potassium channels close very slow, it allows too much potassium to actually get out of the cell. And so that's why we end up with a greater negativity than we should have. Okay, because the potassium channels are too slow to close, too much gets out. Okay, so to recap, at threshold, what's the measurement for threshold? -55, perfect. At threshold, that membrane permeability to sodium will change at the Axon Hillock, which is the trigger zone. And that's going to allow those voltage-gated sodium channels to open. Sodium comes in. We go through depolarization into a very positive state on the inside of our cell. Then, after that, the potassium channels are going to open after those sodium channels have closed. Potassium is going to leave the cell. We're going to go through repolarization to reestablish the resting membrane potential. Of course, when too much potassium leaves, we get the hyperpolarization, and we have to get it to reset with the help of that sodium-potassium pump. So this just happens over and over. Depolarization, repolarization, depolarization, repolarization. This is how the neuron is going to talk to all of the other cells. Is everybody okay with it this far? Anybody have any questions? Anybody lost? I feel like you guys are doing a great job of like staying with it and answering the questions. So if you are good, we're going to keep moving forward. Okay, this is just a recap. So just to make sure that you wrote all of your words in the right place, this is a graph that you can use to check and evaluate. Make sure that you've got it all correct on your study notes. This action potential, make sure you know this is an all or none response. And what that means is we're either going all the way or we're not going at all. So if we hit 55, then that's the signal we're going all the way. Okay, there's no turning back. But if we can't hit 55, then we're just not even going to go at all. We're not going to halfway do something. We're going to do it full out. All action potentials are of the same strength, but not necessarily the same frequency. So if you have a greater intensity of stimulation, for instance, my example earlier was you walk outside and there's like a gentle breeze. If, however, you walk outside and there's a massive tornado and trees are falling all over the place and you're basically being hit in the face by wind, that is going to be a different type of stimulation to your neuron. It's going to tell you, okay, we are in a very big problem. Gentle light breeze, not bad. Massive tornado, we need to do something else. So it's not necessarily the strength that will change, it's the intensity that will cause a different frequency that will make that neuron respond at a different level, so that way your body is taken care of. Okay? We have these things called refractory periods. Now this is a small period of time after an action potential occurs where it doesn't matter what the stimulus is, even if it's strong, it's not going to cause that neuron to go through another action potential again. And this is important for two reasons. Number one is because this limits the frequency of action potentials. If you're always having high frequency action potentials, it would drive you crazy. You wouldn't be able to rest and relax. So this limits the frequency of those action potentials, and it ensures that that impulse only goes in one direction, as we've already said. One direction, down the axon, away from the soma. So that's what the refractory period is going to do. Let's look at this graph so you can see with the dark purple, this is called the absolute refractory period. You can see here that this takes up pretty much the entire time of depolarization and repolarization. The absolute refractory period means absolutely not. Does not matter how strong the stimulus is. It is not going to fire another action potential. Really, no matter what you do, it's irrelevant. Okay? No more action potential in the absolute refractory period. Relative means, well, maybe. Okay? Relative is the big maybe. If we can get a strong enough stimulus, because as you can see, we're down here in the hyperpolarized state. Remember, we have to get to -55 in order to fire an action potential. So it's going to be harder to actually reach the threshold down here than it is if you were sitting at the resting membrane potential. So if the stimulus is strong enough, then yes, you could get another action potential, but it has to be super strong, okay? So that's why it's called the relative. All right, once we have information being transmitted through the axon, then the next thing that has to happen is we've got to go towards the telodendria at the axon terminal. So what you can see here is this particular neuron that's not completely drawn. You can see that it is sending information down this axon, and this little circle is blown up here at the top. And so what you see here is there's a space in between this axon terminal and the next neuron in line. So what I want you to do is I want you to get out your neuron picture, and I want you to write after the axon terminal, I want you to draw an area of space, and I want you to write in there synaptic cleft or synaptic gap. Cleft or gap, whichever one you want, doesn't matter, mean the same thing. This signifies that there is going to be no touching with these axons. Okay, think about them as antisocial introverts. Don't want to be touched. I'll communicate with you, but I just kind of want you to keep your distance away from me. Okay? So the synaptic cleft is very, very important, because what that means is once the information travels down and it hits the axon terminal and the neurotransmitters are released, that means the neurotransmitters have to diffuse across that gap in order to reach the next cell in line. So the diffusion process is going to be very important to ensuring that we can continue to pass along the message. So I'll show you some pictures of that here in just a little bit. Propagation is how we are going to move that action potential along the axon. That's the actual terminology for it, propagation. Make sure that you know the two different types. Type number 1, continuous. This is in unmyelinated axons. Unmyelinated axons. So for instance, if I were to move to my car after I left here, I would have to go through the door, make a right, go down 3 flights of stairs, you know, every single step, one by one by one, walk out the door, go to my car. That's a continuous motion that I would have to make to get from A to B. And it's going to take me some time to do that. So it does take time for continuous propagation. However, saltatory propagation is a whole lot faster. So let's say that somebody made me a jetpack and I opened up this window and I were to jetpack out this window and I get to my car in like as fast as the snap of a fingers. That's saltatory propagation. I don't have to take all of these steps and I don't have to, you know, walk one by one by one. I'm there very, very quickly. So saltatory propagation will occur in myelinated axons because the myelin is kind of like the jetpack. It is what is going to allow that information to move a whole lot faster. So not only will this move faster, this is also going to require less energy. So that's good for body purposes, right? It doesn't take as much energy to actually make this happen. The reason why this can happen quickly is because that myelin is going to have those nodes. Remember, we drew the nodes of Ranvier on your picture. So what's going to happen is the action potential is going to jump from node to node to node. It's kind of like the jetpack thing. You're going to fly over a big, huge portion of the axon. So you're going to be able to get to the end a whole lot faster because you can jump from node to node. So whenever you are looking at saltatory, keep in mind it's only saltatory if that axon has been myelinated, because that's the only way it'll be able to jump, is if that myelin is actually there. Not every axon is going to be myelinated. This is a really good video that I found for you guys. So if you want to watch this video, this is basically a recap of everything that we've talked about up to this point. So if any part of it is confusing to you, then I highly encourage you to go back and watch that video a couple of times. It's only like 5 or 6 minutes long, but really good at summing it all up. Okay. The other thing that can affect the axon diameter, the other thing that can affect propagation is the axon diameter. So the larger diameter you have, the faster the speed will be, because there will be less resistance that is going to be met whenever that information is trying to move down the axon. So large diameter, fast speed. We have 3 different types of axons, and we classify them based on their diameter, if they're myelinated, and then how fast they are. So A, B, and C are 3 different types. A is the fastest one. This is myelinated, of course. It's got to be fast. Very large diameter. So circle 120 meters per second. That's how fast that information can move, 120 meters per second. So very important things that are going to, maybe possibly threaten your life are going to have to run through type A fibers so that that information can be transmitted rapidly. Type B is myelinated, but it's only a medium diameter, which means it only goes 18 meters per second, which is significantly different. And then type C is unmyelinated and small, so it's 1 meter per second. And that's going to be mostly sensory information. So all of these messages are carried according to priority. The body will always do things in priority. So whatever is most important is going to be transmitted through that type A. Like one day I was driving home and it was 9:30 at night and I was looking at my phone because I was trying to find a particular song on Spotify that my lab class had just got finished talking about, and they really liked the song, and so I was trying to find it for lab, and I looked up and I saw in front of me, not even that far in front of me, a couple hundred feet, a car driving the wrong way down this one-way street. and it was headed directly for me. It was a 2-lane road, but we were in the same lane. And so my type A fibers kicked in fast enough for me to yank the car so that way I could get out of the way. And thank goodness my car has really good handling. I didn't run up on the sidewalk or anything. I didn't damage my car. But as soon as I moved over into the other lane, that car zoomed right by me. So I'm sure he was drunk, but it was in that day that I was like, oh, thank God for my A fibers. So, you know, we don't know about these things until we learn them. And then we're like, oh, I'm so appreciative for that, right? So it just saved my life. Anything that threatens your survival is going to run through an A fiber. All right, now the synapse. We've got to talk about the synapse because, of course, once all of the information gets transmitted from the dendrites through the soma, down the axon, down the telodendria, we got to go to the synapse. So if we're looking at a presynaptic neuron, just know that that means we are talking about the neuron that is actually sending the message. Okay, presynapse, it's before the synapse. Postsynapse means this is the one that's going to actually receive the message. So it's going to be on the opposite side of that gap or that cleft. So we'll have a presynaptic neuron sending, a postsynaptic receiving. Now, where is the postsynaptic neuron going to receive that information? What part of the neuron? The dendrites, right? So the dendrites are going to receive that message. And it's going to say, okay, what all is going on? What do we need to do? There's 2 types of synapses. Now, I told you guys to draw that space on your picture. So I want you to go back to that little space of the synaptic gap or the synaptic cleft and make sure that you write, this is a chemical synapse, okay? The chemical synapse is the one where the neurons don't touch. Very rarely do we have the electrical synapses where we do have neurons that are touching. These are the neurons that are not antisocial introverts, but it's very rare. So you can see the electrical synapses, the pre and the postsynaptic membrane will be locked at gap junctions. So because they're locked together at gap junctions, then that means that this information is going to be able to pass through the gap junction instead of having to diffuse across a space. So it's going to be moved very quickly and it's going to be pretty efficient. Okay? But our chemical synapses, this one with the cleft, this is the most common one. So this is why we're going to talk about this one because this is the one that you're going to see most often. You can see here, this is a picture. This is the presynaptic axon terminal. Synaptic vesicles inside of them have neurotransmitters. Okay, so that's what you're looking at right here. You see the space, and then you see the postsynaptic membrane of this neuron down here. We have 2 types of chemical synapses. We have neuromuscular, obviously as the name implies, between a neuron and a muscle, neuroglandular, between a neuron and a gland. So those are pretty self-explanatory. The neurotransmitters, star, 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 these are the chemical messengers. They are the ones that are going to transmit the information that is being moved along from neuron to neuron or from neuron to cell, whatever is happening. The chemical messengers, the neurotransmitters, are the ones that are going to pass that information down the road. So very important that we know about neurotransmitters. They are the ones that will be released into the cleft and that will have to diffuse across to that postsynaptic membrane. They will be broken down by enzymes that are within the synaptic cleft. It's very important that that happens so that then they can be reabsorbed or reassembled by the axon terminal. And I'm going to show you how that happens. The function of chemical synapses, obviously, we are going to be looking for potentials. We want some action potentials so that we can actually pass messages along. That's the whole purpose here. So if we're looking at a cholinergic synapse, this is going to release acetylcholine. Make sure that you know acetylcholine is ACH. because you guys will talk about this through 2401, talk about through 2402, so you cannot forget acetylcholine because it is so common to learn this particular neurotransmitter through AMP. Okay, since all neuromuscular junctions are going to use acetylcholine, this is why we definitely talk about it in 2401 as we learn the nervous system and the muscular system. But you can see it also happens within the CNS, the PNS, and even the ANS, the autonomic nervous system. So very important neurotransmitter. For my words people, this is your slide right here, but I'm going to do the picture, the 4 pictures that follow to kind of talk about what's happening in this synapse, so that way you can kind of see and understand with the picture. So we can see step number 1, we have the action potential, depolarizing the axon terminal. So you can see this action potential is coming down. When this action potential comes down, it's a signal that it's time for the axon terminal to get ready to do some work. So what work does it need to do? It needs to open these voltage-gated calcium channels and allow the calcium to flow in. When this is depolarized, these channels will open and calcium can come in from the extracellular fluid into the axon terminal, and that is the trigger that's going to tell the acetylcholine it's time for you to go. Okay, so how is it going to go? Through this word, exocytosis. Exo meaning exit. So the exocytosis is what you see on this part of the picture here. It's leaving the axon terminal and it's going to have to diffuse across to this receptor on this postsynaptic membrane. Write in the word receptor right here since it's not here, because this becomes incredibly important in the information transmission. If the acetylcholine comes out and there's no receptor for it to dock onto, well, now we have a problem. Okay, we got a big problem. So the receptor is very, very, very important. So what you see here, as the acetylcholine is released through the exocytosis, it's going to come and dock on the receptor. Once it docks on the receptor, it's only going to be there for a few milliseconds, but this causes those chemically gated sodium channels to open. Remember, if we move sodium in, then we change the actual measurement inside of the cell in a more positive direction. If we can get to -55, then we'll get an action potential. If not, then it's just going to remain a graded potential. So once that acetylcholine has done its job and it has been received by the receptor, we have this very important enzyme here, this acetylcholinesterase, that is going to break the acetylcholine apart into 2 pieces. It'll break it into an acetate and a choline. So just circle right here that the choline goes back inside of the terminal. The choline has to be recycled so that it can be reused. The acetate goes off and it's absorbed in the extracellular fluid. But the choline needs to come back in. Now, although this seems like, okay, yeah, sure, that's great and wonderful, you don't realize how important this enzyme is to you until you do not have its full function anymore. So what I want to show you real quick, let's get a new share. Okay, this is an axon terminal. This is the postsynaptic membrane with its receptors. We've got to release the acetylcholine, but we need acetylcholinesterase sitting here in this cleft to break that acetylcholine apart when it's time. There are things that are called competitive acetylcholinesterase inhibitors. If you inhibit something from doing its job, what does that mean? Are you stopping it?
Yeah.
Or are you helping it? If you inhibit, stopping it, right? So if you stop something to inhibit it, it means you stop it. If you stop that enzyme from breaking down that acetylcholine, then that becomes very toxic to your body. There are chemicals called organophosphorus compounds. They're also commonly known as nerve gases that will stop the acetylcholinesterase from doing its job. So what that means for you is, you can see down here, as little as 1 milligram of these compounds will cause profuse sweating, dimmed vision, uncontrollable vomiting and defecation, convulsions, filling of the bronchial passages with mucus, bronchial constriction, and at last paralysis, and you will die from choking and not being able to breathe. Okay? All because of these compounds that used to be used as insecticides until they realized how horrendous these things were. Now, there's a lot of pesticides and herbicides that are out there right now that are very horrendous. They just don't have these immediate effects, but they're still bad long term. So if you do not know what they are and you do not know what foods they're used upon, you need to definitely be doing some work into that because you might be eating some of them, okay? So it is possible to get some drugs that will help reverse this, but you have to be fast enough in order to get the antidote that works against the inhibitor in order for the acetylcholinesterase to go back to doing its job like it's supposed to and breaking down your acetylcholine as it should. So you do not want an accumulation of acetylcholine in the synapse because it will be so toxic to your system. So I know you didn't appreciate acetylcholinesterase before you got in here, but I bet you do now, huh? Okay, so very important enzyme. We got to have it. We got to let it break down that acetylcholine so that our body works as it's supposed to. Now, there is such a thing called the synaptic delay. This is normal. This is basically the time that's needed for that calcium to come in its own voltage gated channel to signal the release of the neurotransmitter. Okay, so that's common. And then we also sometimes will have synaptic fatigue. This occurs when the neurotransmitter cannot be recycled fast enough to meet the demands of any intense stimuli. So the response of the synapse will weaken until the acetylcholine can be restored and replenished. We have different types of neurotransmitters. Excitatory means that they cause the depolarization. So they're going to actually help make action potentials. Inhibitory, remember, that means stop. So they're going to suppress the action potentials. Now, there can be tons, as we've seen on some of the pictures that I've showed you. We had a couple of different axon terminals coming into just that one neuron in some of our pictures. There can be tons of neurotransmitters that are received by just one neuron. And even though there may be thousands of neurotransmitters that are received by that one neuron, it's going to be the summation of all of them together. Some may be excitatory, some may be inhibitory. It's going to be the summation and the net total that says, okay, what should I be doing? Should we be doing the action potential or should we not? So that's why that Axon Hillock is very important, because it will determine whether or not that action potential needs to actually go or if it needs to slow. Okay? Because we don't have a lot of time, I'm just going to tell you to go back and watch this YouTube video on your own. But everything that we just talked about in the synapse, from the diffusion of the neurotransmitters to the receptor to the enzyme in the gap that will break neurotransmitters apart, all of that is what is basically utilized whenever it comes to drugs, whether those are legal drugs or illegal drugs. But that video right there will show you what happens to a person when they take heroin or when they take cocaine or when they take methamphetamine. What are they actually doing to their neurotransmitters? And then what's actually happening to the receptors? Because you know, the more drugs you take, the more your body starts to adapt. And the more your body adapts, the more you throw off your neurotransmitter balance. The more you do that, now you also start impacting the receptors negatively. You could even start losing receptors. That's why they need more and more and more and more drugs, and then overdoses happen, which is horrible. So that video is really eye-opening on how important the neurotransmitters are to stay in homeostasis, especially for people with drug addiction. Now, I put this website on here because you can see that CBD has the ability to work with different receptors, whether it's pain receptors or different neurotransmitter receptors, in order to help get people off of drugs. So it is something that is really remarkable whenever it comes to brain health. And there's a lot of things that people can do with CBD. So if you're not familiar with it, or if you are that person, or maybe you know a person who kind of has a stereotypical view of medical marijuana, I highly encourage you to look a little bit deeper at what the research shows in regards to how positive CBD can really be in people's lives. And I have an assignment for you guys to do that as well. So that way you can really kind of understand a little bit more about it. So a couple of things I want to leave you with. Star this: the effect of a neurotransmitter depends on the property of the receptor. That receptor is really going to be the one that reads it and says, this is what we're going to do. So it's very important that we understand how receptors are really going to be big keys here in what's going on with these action potentials. Neuromodulators are chemicals that can be released by the axon terminal that will change the rate of neurotransmitter release or the response by the postsynaptic cell. So some neuromodulators, like opioids, will cause you to not feel pain while you're going through a very intense situation. So like in domestic abuse, if a woman is being beaten, she usually does not feel pain at that point in time, because as the neurotransmitters are all going through her body, she can also have these neuromodulators that help kind of keep her away from the feelings of pain. It's not until she leaves that situation and realizes how bad everything is, and then the pain starts to kind of flood in, because now those neuromodulators aren't there anymore. But this is also like those endorphins that allow moms to get their kids out of really bad situations really fast. So these neuromodulators really kind of help the body in multiple different ways. Obviously, they can be misused, right? Because if a person starts feeling the effect of opioids, then they want it all the time, and then that can lead to some, drug addiction things, obviously. So that's why I also have the CBD thing in there for you guys to learn about. We also have gases as neurotransmitters. As weird as that sounds, nitric oxide and carbon monoxide can be used as neurotransmitters. I talk more about the NO in 2402 when we get into the cardiovascular system, because of its role in that system. Especially important to understand how that works with diabetic patients. So we'll talk about that a little bit in 2402. Here is charts of different types of neurotransmitters. You do not have to memorize all of these things, but it's just so that you can see acetylcholine, norepi, epi, dopamine, serotonin, all these different neurotransmitters, and basically how they're going to help maintain homeostasis inside of the body. Okay, so here's your assignment. Obviously, it's pretty self-explanatory. Just make sure that you watch this video, read these 2 articles. If you want to look at more, read more, by all means, feel free to do that whenever you decide that you're going through this. If you see something that catches your eye, if you know some personal situations of people, then, if you're thinking about people, look for them. Find some research for them of things that may be helpful and beneficial to them as you go through this, because that's what we're doing it for, is to help other people. All right, anybody have any questions? No?